which looks like which that would look like. This is a red blood cell, right? Whereas dad's or mom's, the parents' red blood cell would look like that. Okay. So they're saying that if a male parent is the one that's type O, dad, it would be impossible to have an AB because it's codominant, right?
Mhm.
Unless dad is not really O. Dad very well could be A, B, and he could be this, or this. Wait, I should go in here. Or he could even be this. Right?
Okay.
But he's reading as type O, because what he doesn't have is this thing, let's just call this our antigen binding protein.
Okay.
This A and B are your antigens, and this is your binding protein. And if he happens to have another gene that codes for him not to have this, genetically, he could be an A or a B or an AB. But phenotypically, he's going to express as an I, because these A's or B or AB are off floating around, but not actually attached to the red blood cell. So genotypically, he comes out as an AB, or genotypically he comes out as a type O, but genotypically he comes out as an A, a B, and AB, which would mean he could have an AB child, but phenotypically he comes out looking like an O. That's the Bombay effect.
Because it's not attached.
Yes, it's not attached,
Okay.
even though genetically it's there. So genetically, when that genetic code copies, it's going to copy into the offspring and give them an A or B blood type,
Okay.
whatever happens to be. Just phenotypically, he won't express that way.
Okay.
And so you're thinking, oh, that's not my kid. Totally the
Yeah.
kid. So that's to make you remember that. And then we look at the RH factor. We remember RH factor being a protein that's on the red blood cell, right? If you have it, you're Rh positive. If you don't have it, you're Rh negative. So then we look at blood types in a new way, with an antigen A, antigen B, or an antigen, and both antigens A and B, and you either are Rh positive, or Rh negative, meaning you don't have that protein whatsoever. You should also consider, let's see, what the universal donor is. O, because you could be with anybody else's blood type because you don't have any antigen that would be recognized as non-self. Universal acceptor is A, B. They can accept A, they've got it. They can accept B. What?
In the Bombay effect right there, if I was to get blood from the O, but it also A and B antigens, is that going to work or no?
No, because you're still going to have the. You know what? I should actually look that up because technically you'll still have those antigens in your
yeah.
system,
Yeah.
and it should recognize you as non-self.
Okay. So it will attack the AB, but not the blood
But
cells
possibly,
still.
yeah, not the blood cells.
So we'll still be able to get the blood though.
Possibly. I'd actually have to look into it to find out what happens physiologically, because I'm not sure. Okay, so what you're looking at, okay, so universal acceptor, AB, because they recognize A, they recognize B, and if you have none of those, it recognizes that too, and that's fine. It's acceptable. You should also consider that with RH, when we go over one of these questions, RH negative versus RH positive, if I'm RH negative mom, I have my first child, and during birth, things get a little bit messy, bloods mix. And immediately, when your blood mixes at that time with your kid, and your kid happens to be Rh positive, your blood starts making antibodies immediately. So when you get pregnant again, and that child's Rh positive, your body immediately recognizes it non-self and tries to get rid of it. So you have to go on immunosuppressive therapy if you're an Rh negative mother with an Rh positive child for the second kid. You should also take a look over at your, you should also take a look over at your notebook, mine should be somewhere, for your different types of ratios for expected phenotypic and genotypic outcomes, the 9:3 to 3:1 ratio for a dihybrid cross. Pardon?
I remember.
Okay, and you should consider remembering linked traits, the red hair and freckles. If you have freckles, you're going to have, or no, if you have freckles, you're going to have red hair. If you have red hair, you don't, no. If it's red hair, you're going to have freckles. If you have freckles, you don't necessarily have red hair.
Oh.
But they're linked together. Okay. DNA profiling, or when we did this is where we did the dye gel to prep you for actually doing restriction enzymes. We want to do, if you're going to get it wrong here, and not get it wrong here, this is more expensive. So the equipment that you use were gel boxes and micropipettes. You should remember how to use your micropipettes. You should definitely remember how to use your micropipettes because you're actually examined on it.
Yeah.
They're not.
Okay.
So remember, push in before we turn? Yes, because otherwise you're gonna break it. And then for your specific things, you will be totally disqualified on the exam immediately if you crank it, yeah.
Okay, without pushing it?
Yeah, so just FYI.
Okay.
Okay, so in this we used dye, and it was a mixture, and we put our comb in the center of our gel so that we could separate it according to size, charge and size. Actually, this one we only separated it according to charge. DNA is size, charge and size. This one we only separated it according to charge, and we found that we had complete, we had pure mixtures, and then we had pure dyes, and we had mixtures of dyes. Which separated out, you might want to go and review your pictures that you took.
Yeah.
Chemicals we used were buffer, which we filled the wells of our electrophoresis box. We used TAE, triacetic acid, EDTA. We ran it at approximately 150 volts for our 30-minute run time. The medium we used to separate this out in was called agarose. We'll remember that, let's see, anything else that I can think of? You should remember actually how to assemble your gel box. Yeah.
The TAE was just used to provide a medium for the current, right? That's what.
For electrical current, yeah.
Okay, that's
We use TAE as a buffer or we use TAE as a current for current to run through it. In the DNA Inquirer, we use the same equipment. We use a gel electrophoresis box. We set up our cassette, raised our dams, poured our agarose, dropped our dams, placed it in the box, filled the wells with our TAE. Then we put in our combs. No, put in our combs after we put in our combs after we filled it with the agarose. Then we filled it with the TAE. Then we pulled our combs. And if you wanted to practice pipetting, you probably pipetted into those empty wells to clear out any of the extra jello. Then you started your actual pipetting. Yes.
There were two buffers used. One was TAE and there was another one.
The other one was restriction buffer.
Okay.
The restriction buffer was used in your enzymes so that they did not denature
Okay.
due to changes in pH level. We kept everything super cold because, again, small amount of time, we've had denaturation. The enzymes we used were HIND, ECO, and BAM. These are to cut at specific sites of base pairs.
I remember.
Pardon?
I remember how many cuts they make on from the DNA formatting.
Okay, and so what was our control? DNA, no enzyme, so we have no cuts. We used a hot bath to accelerate the cutting from the ECO, HIND, and BAM. And we used Easy View dye after we did all the cutting so that we could actually see. We used calcium chloride, no, we did not use calcium chloride, this one was like we used restriction buffer in the little micropipette tubes, and the DNA we cut was lambda, so it was Foch bacteria. We ran our currents, and we went in only one direction, so we started our gel at the very top to separate out our DNA through size, and we ran it for approximately an hour at between 100 and 110 volts. And then you should review how things could possibly go wrong. Look up online. Look up online different gels. They'll show you get many pictures of gels that ran too hot, ran too short, ran too long, to see how things might look funny. If you were to give it a random picture, or maybe look at your guys' gels from class to see just how things might possibly go wrong for you. And that if you were to be given a picture of it, like I did on one of these over here, you could actually give me an answer, yeah, that one, of how things might have gone wrong. That one probably ran too short and too hot. The bands are super close together, and it looks like a kind of a fireball came through, and I almost think that somebody stabbed a well over there too, because you see phosphorescence underneath the wells. And if DNA runs all the way through, it should have run all the way through and not stick under a well. So when we go over to DNA isolation, this is where you grab out stock from a container, essentially. DNA isolation, we use the equipment, the blender. I chopped up your onion for you, but you would have chopped it if I had it. You added it to a tube. You added that with detergent. The blender, then you mix it together. The detergent breaks down the cell membrane. The blender helps to break up the cell wall. The meat tenderizer in the detergent helps to break down the histones, the proteins that help bind up the DNA. After you did that, you straightened out all the solids because you don't want those. And you poured a small, thin layer of ice-cold ethanol over on the top of this kind of slurry, at which point the DNA precipitated out. It looks like little wispy strands due to the polarity. That's why you used alcohol, ethanol. And then you use methyl blue, that was your stain, to dye your DNA for what you looked at it, and it looked like just big strings because it looked anticlimactic. But the whole exercise is to show that, that you can actually pull your DNA out, and to show you, it was kind of like a quick and dirty method, that you can actually do this, and then you can actually even amplify it, and then run gels later, which is what we did in the
Okay,
beginning.
we could amplify it too without PCR.
You do that with the PCR.
Oh, you had to use a PCR.
Yeah. Okay, then we did bacterial transformation. So you have normal bacterial cells for which you have DNA in them. You have normal bacterial cells that have DNA, and they have extra chromosomal DNA called a plasmid. These naturally exist. What we use is E. coli. It's the one that most people, and when I say people, I mean bacteriologists, it's the most easily manipulatable. So we often use E. coli. So what you guys did was you took totally normal cells and you transformed them, taking a piece of a plasmid from another cell and introducing it into this cell by way of calcium chloride. So what you did is you used E. coli bacteria as your choice of bacterial cell for manipulation. We used a few different things, a few different chemicals. We had lauria broth, which was the food for our bacteria. We had pea green plasmid, which was the protein, that was our protein production instructions. And it also gave us ampicillin resistance instructions. We used calcium chloride, which makes our cell membrane permeable. And we had our plasmid, of course, which is what we needed to put in. Okay, so what we did was, I'll erase this. You took your normal cells of E. coli, you added calcium chloride, which gave you permeable cells, plus you added plasmid, and what you ended up getting was transformed cells with this plasmid. Not every cell gets transformed. So what we gave you were these different plates. We gave you ampicillin plates on agar, or agar impregnated with ampicillin. We gave you agar with no ampicillin. And what you did was you took these newly transformed cells and you put them on an ampicillin positive plate with no plasmid, an ampicillin positive plate with plasmid, a non-ampicillin plate with no plasmid, and a non-ampicillin plate with plasmid. And the outcome is expected. On an ampicillin plate where you have no plasmid and you lay down bacteria, it's going to die. Ampicillin is going to kill. It's a beta-lactam antibiotic. Pardon me?
Just kill the bacteria.
Yeah, it will. Yeah, by breaking the NAG down chains, and it causes it to lyse. Okay, so expect that they're all going to die. You have a one dead. And then on your one with plasmid, which you had these bright green colonies, and then you had milky colonies on the outside of it, satellite colonies. And that is because any of the bacteria that happen to be transformed would release a protein, two proteins in fact. They'd produce the protein that made them green, and they'd produce a protein that, through diffusion, would go ahead and break down the ampicillin. We'll call ampicillinase as an enzyme. As a result, any bacteria that were super lucky to land within the direct vicinity of those transformed bacteria were benefited by the fact that the transformed bacteria broke down the ampicillin in that direct vicinity, and therefore they were able to live and colonize. So you have both transformed and non-transformed bacteria able to coexist. In the one without plasmid and without ampicillin, you have a full lawn growing because you didn't put a plasmid, but you have a bacteria, and you gave the bacteria food, and it grew. No surprise. The one with plasmid, you equally had a lawn, and you may or may not have been able to see little green dots in it because it's like looking at a green grass and trying to see if there's yellow specks. Or looking at a yellow lawn of milky E. coli and trying to see if you have little transformed green specks. It's hard to see, but they are there because the majority of your bacteria are going to be non-transformed. There are going to be a few that are transformed, and those few that are transformed are going to be outcompeted by the normal E. coli because in an environment where there is no ampicillin, it is counterproductive to produce a protein that you don't need.
Which is small.
Pardon me?
They're probably small too.
Well, yeah, they're going to be single cells.
Yeah.
So, but I mean, and they're going to be outcompeted just because, not because they're small. I mean, because if you put those plasmid ones in an area where there was ampicillin, these ones, they were bigger, they grew hardier. And it was only by luck of the fact that the ones that weren't transformed that landed near the ones that were transformed that they were able to even survive. Here, however, they're busy pumping out a protein that they don't need because they're not in a toxic environment. So it's going to cause them to grow slower. Whereas these guys don't need to pump out that extra protein. They can put all their energy into growth, and so they do, into a giant lawn. So this is where I would start if I were going to be studying for this exam. I would just go lab by lab and data dump as much information out as I could. And then even for myself, well, I feel like I've covered this, I would go back I know I very briefly did this with you, telling you that, you had your DNA, you had your E. coli cells, you put them with the calcium chloride, you put them with the plasmid,